This is a talk about fossils, what they are, what they're not, and why they're much more than just attractive objects in rocks. Fossils are our main direct evidence for the history of life. They let us see organisms that are long since extinct, ecosystems that no longer exist, and evolutionary changes that took place millions of years ago. However, the fossil record is incomplete and selective. So I'm going to answer two basic questions. The first is, what are fossils? And the second question is, how do fossils form? Most organisms don't become fossils when they die. They decay, they're eaten, they're broken up, or they simply leave no durable trace. So fossilization is unusual, and the fossil record is shaped by a series of filters. Before we talk about fossils themselves, it's worth remembering that fossils occur in rocks, and that not all rocks are equally good at preserving them. Geologists usually divide rocks into three broad types. Igneous rocks form from molten material, magma underground or lava at the surface. Examples include granite and basalt. These rocks are often very important for dating geological events, especially volcanic ash beds, but they're not usually where we expect to find fossils because the heat involved in their formation is generally not kind to biological remains. Metamorphic rocks are rocks that have been altered by heat and pressure, but without completely melting. Marble, quartzite and slate are familiar examples. Fossils can sometimes survive low-grade metamorphism, but the more intense the heating and deformation, the more likely original biological detail will be distorted or destroyed. Most fossils are found in sedimentary rocks. Sedimentary rocks form when particles such as sand, silt and clay accumulate, are buried and become cemented together, or when minerals precipitate chemically from water. Sandstone, shale, limestone and dolomite are examples. Because sedimentary rocks form at or near the surface, often in rivers, lakes, seas and deserts, they're much more likely to entomb organisms or traces of organisms. So what is a fossil? A useful broad definition is that a fossil is any preserved evidence of past life. We can make a basic distinction between body fossils on one hand and trace fossils on the other. Body fossils preserve some part of the organism itself. These might be hard parts such as bones, teeth, shells or calcified plates. But body fossils can also preserve softer tissues in exceptional circumstances. These are much rarer, but they're often especially informative. Sometimes we find remains of skin, muscles, guts, feathers, eyes, or even the outlines of organisms that had no hard skeleton at all. These fossils are important because they remind us how incomplete the fossil record usually is. Trace fossils are different, since they preserve something that the organism did. Footprints, trackways, burrows and borings, feeding marks and resting traces, fossil feces and root traces are all examples of trace fossils. They can be extremely useful because they record behaviour and they can tell us how animals moved or where they lived, how they fed, how they interacted with sediment, and sometimes how they interacted with one another. At a still finer scale, chemical fossils or biomarkers can preserve molecules, isotopic signatures, or other chemical evidence of biological activity, even when no body fossils remain. Most fossils, however, are hard parts: bones, teeth, shells, carapaces, woody tissue and other structures that already have some durability before they're buried. This matters because the fossil record can look more complete than it really is. What we see is not simply what once lived, it's what once lived and then had the right anatomy and the right environment and the right geological history to be preserved. So how do fossils form? In the sea, at least, the basic sequence is this. First, the animal dies. Almost immediately, biological processes begin to work against preservation. Soft tissues start to decay, and bacteria attack these. Scavengers can eat parts of the body. Water currents and other animals can all break up or scatter the remains. The key step for fossil formation is usually rapid burial. If the skeleton or shell is buried in sediment, it's protected from some of the processes that would otherwise destroy it. Burial reduces disturbance by scavengers and physical abrasion, and it may also reduce exposure to oxygen, and this slows down decay. Later on, more sediment accumulates above it, and as that sediment builds up, it compacts and squeezes water out, pushing particles closer together so that minerals can begin to precipitate between the grains. This is part of the process by which loose sediment eventually becomes sedimentary rock. The fossil itself is also usually altered. The original hard parts may remain, but often they are recrystallized, replaced by other minerals, or dissolved away to leave a mould or filled in to form a cast. So a fossil is rarely the original material of the organism. Usually it's a geological copy of its shape. There's another important point here. Fossilization is not necessarily a gentle process. Compaction can flatten or distort the remains. Chemical change can remove detail. Later tectonic movement can fold, fracture, uplift or expose the rocks, so that by the time we find a fossil, it's usually passed through a long sequence of biological, sedimentological and geological filters. The last stage is exposure. A marine fossil can end up exposed on land because the rocks that contain it have been uplifted, tilted and eroded. And that's why we can find the remains of marine animals in cliffs and mountains far from the sea in which they originally lived. So fossilization is a chain of events: death, decay, burial, chemical alteration, rock formation, uplift and exposure. Break the chain at almost any point, and the fossil is never found. Counterintuitively, the manner in which the organism died can sometimes give us additional information. Tooth marks, punctures, crushed bones, drill holes in shells, healed or unhealed injuries, and patterns of breakage can all suggest predation, scavenging, disease, or physical trauma. The important caveat is that the same mark can sometimes have different causes. A tooth mark made by a predator before death can look very much like damage made by a scavenger after death. Death can also add information about place. Where did the organism die? Was it in a desert, a river channel, a lake, a mud flat, a shallow sea, or indeed the deep ocean? The surrounding sediment is often crucial here. Mudstones can suggest quiet water. Cross-bedded sands can suggest currents. Ripple marks could indicate shallow water. Evaporites may point to arid, saline settings. Volcanic ash can suggest rapid burial after an eruption. But we have to ask whether the fossil died where it's found. Some fossils are preserved close to where the organism lived. Others were transported after death. A shell can be washed along a beach, or a carcass can float before sinking. Bones can be moved by rivers, waves, currents, or scavengers. The accompanying fossils are another source of information. A fossil found with corals, brachiopods and crinoids tells a different ecological story from one found with freshwater fish, land plants, or dinosaur footprints. Associated fossils can help reconstruct habitat, water depth, climate, and indeed community structure. They can also suggest whether we're looking at an ecological community, a transported assemblage, or a time-averaged mixture of remains accumulated over many years. A puncture mark in bone can record a bite, but was the animal attacked while alive or scavenged after death? If there's healing around the injury, it shows the animal survived for some time after the damage was inflicted. Lucy is a different kind of example. Lucy is the famous partial skeleton of Australopithecus afarensis from Hadar in Ethiopia, about 3.2 million years old. She's one of the most important hominin fossils because a substantial part of her skeleton is preserved, and this tells us a great deal about early bipedalism. But there's been a debate about how she died. In 2016, Kappelman and colleagues published a paper arguing that some of Lucy's fractures were consistent with a fall, probably from a tree. They suggested that she may have landed feet first and then thrown out her arms, producing fractures. However, other paleoanthropologists have argued that some or all of these fractures could instead reflect damage after death: fossilization, compaction, pressure in the sediment, and breakage during exposure. So Lucy is useful here not because we can say with certainty that she fell from a tree, but because she shows the kind of reasoning involved. We compare the pattern of damage with modern forensic cases, and then ask which explanation best fits the whole pattern. Taphonomy is the study of the processes between death, decay, burial, fossilization, exposure, and ultimately fossil collection. Information is certainly lost, but information can also be gained. Once an organism dies, it enters a particular sedimentary and ecological context. For example, a living animal doesn't carry a label saying lived in shallow marine mud, but after death, if it's buried in fine laminated muds alongside marine bivalves, burrows and wave ripples, we can infer something about the environment in which it was buried. Similarly, a shell drilled by a predator can preserve evidence of an ecological interaction that would otherwise have gone unrecorded. Taphonomy is therefore a filter, but it's not just a destructive filter. It transforms biological information into geological information. It removes some signals, distorts others, and adds new contextual evidence. The skill is to separate the original biological signal from the taphonomic overprint when we interpret this. So what influences the rate of decay? Well, this depends on the organism, the environment, and the chemistry of the sediment. This is one of the reasons why fossilization is so selective. The first major factor is oxygen. Aerobic microbes can break down organic matter very efficiently, producing carbon dioxide and water. This is energetically favourable, so aerobic decay is usually very quick. It's also why oxygenated environments are often bad for preserving soft tissues. But aerobic decay consumes a huge amount of oxygen. If a carcass is buried quickly in fine sediment, or if the surrounding water is poorly mixed, oxygen can be used up faster than it's replaced. Once the immediate environment around the remains becomes oxygen poor, decay doesn't stop, but it changes. Microbes can use other electron acceptors such as nitrate, iron, manganese, sulfate or carbon dioxide. These anaerobic pathways are often slower and produce different chemical conditions around the carcass. Now, this is important because the chemistry around a decaying organism can become very localized. The carcass can create its own microenvironment, a small chemical bubble within the sediment. That microenvironment can influence whether tissues collapse, whether minerals form, whether phosphates, carbonates or pyrite precipitate, and whether fine anatomical details are lost or retained. The second factor influencing decay is temperature. Warm conditions generally speed up microbial metabolism and chemical reactions, so decay goes faster. Cold conditions slow bacterial and enzymic activity, which is why refrigeration, freezing, high altitude and polar settings can all retard decay. The third factor is pH. Very acidic or very alkaline conditions can inhibit some microbes and enzymes. But pH can also damage particular tissues or hard parts. Acidic conditions can dissolve calcium carbonate shells. Alkaline conditions can affect other organic and mineral components. So pH does not just affect the speed of decay, it also influences which parts of an organism are selectively preserved or destroyed. Moisture matters as well. Microbes generally need water, so very dry environments can slow down decay and favour desiccation or mummification. At the other extreme, waterlogged environments can reduce oxygen supply, especially in fine-grained stagnant sediments. This is one reason why bogs, lake bottoms and quiet marine muds can sometimes preserve organic remains unusually well. The nature of the sediment is also important. Coarse sands tend to allow water and oxygen to circulate, so remains can be more exposed to oxidation and disturbance. Fine muds can seal remains more effectively, limiting oxygen diffusion and physical disruption. Sediments can also supply ions that promote early mineralization. In some exceptional fossils, decay itself helps create the chemical gradients that allow minerals to form around or within tissues before they're completely destroyed. Finally, the nature of the organic material matters. Not all tissues decay at the same rate. Soft organs and muscles usually decay very rapidly. Collagen, chitin, cellulose, lignin, keratin in hair, enamel in teeth, bone mineral and shell material are more resistant in different ways. This is why plants, insects, vertebrates and shelly invertebrates enter the fossil record differently. The fossil record is therefore biased not only by where organisms lived and died, but also by what they were made of. So there are different levels of preservation. The best level is the rarest and most informative, preservation of labile soft tissues. These are the structures that normally disappear very quickly after death. Muscles, skin, internal organs, eyes, gills and similar structures. For these to be preserved, mineralization has to happen very quickly, before decay has destroyed the anatomy. The Santana Formation of Brazil is a classic example, with fishes preserving muscles, integument, connective tissues and eyes. The next best level is where soft tissues themselves have largely gone, but they've left an outline. Mineral formation, staining organic films or sedimentary impressions can capture the external shape of the animal before the sediment is fully compacted. Now, although this doesn't preserve internal detail, an outline can tell us about body shape, things like fins, skin margins, fur or feathers, or the presence of soft structures that are not obvious from the skeleton alone. The third level occurs when decay has gone further down the road, before mineralization gets established. In that case, the most delicate soft tissues disappear, but the more resistant organic materials can persist. Chitin, lignin, and cellulose are good examples. This is why arthropod cuticles, plant tissues, spores, pollen, and woody material can sometimes be preserved even when things like muscles and organs are long gone. But the fourth level is by far the most common. Here only the heavily mineralized skeleton remains. Shells, bones, teeth, things like echinoderm plates or heavily mineralized cuticle, crab carapaces for example. These fossils can still be extremely useful, but they offer only a fraction of the original organism. And they also, as we've seen, bias the fossil record strongly towards animals and plants with the most durable parts. These two fossils show why exceptional preservation can change our understanding of extinct animals. The upper example is an ichthyosaur. If we had only the bones, we'd still know that this was a large marine reptile with a long snout, powerful jaws and paddle-like limbs. But the soft tissue outline tells us much more. It shows us the body profile, including structures that are not preserved as bone. In ichthyosaurs, soft tissue outlines have been important in showing the presence of a dorsal fin and a tail shape that made them look much more like fast-swimming fishes or dolphins than like a conventional reptile. That's a major biological insight, not just an aesthetic detail. The lower example is Darwinius massillae, a famous specimen nicknamed Ida from the Eocene Messel deposits in Germany. It's an extremely complete, articulated primate skeleton with a soft body outline and preserved contents of guts and what it had been eating. Messel is a good example of a Konservat-Lagerstaette, a deposit with exceptional preservation. Fine-grained and oxygen-poor lake sediments allowed skeletons, skin shadows, fur, feathers, gut contents and other delicate features to survive in some specimens. So all of these levels of preservation can be understood as a race between two processes. On one side is decay, which progressively removes biological information. First, the most labile soft tissues, then the more resistant tissues, and eventually all but the hardest parts. On the other side is mineralization, which can lock information in place before it disappears. If mineralization happens very early, even muscles, eyes, gut traces, and other delicate tissues can be preserved. If it happens later, we may retain only a body outline, carbon film, or resistant tissues such as cuticle. Later still, most soft anatomy is gone, and the fossil record is reduced largely to bones. So the level of preservation reflects the timing of preservation relative to the timing of decay. So which organisms are most likely to fossilize? Well, in general, marine organisms are more likely to fossilize than terrestrial ones. This isn't because things living in the sea are intrinsically more preservable, but because marine environments are often better places for burial. Sediment is continually accumulating on seafloors, especially in quieter settings like shelves, lagoons and basins. This means that dead organisms can be buried very quickly. On land, the situation is often less favourable. Carcasses are usually exposed to weathering, scavengers, trampling, drying and erosion. However, terrestrial organisms can fossilise very well in particular settings, such as river channels, floodplains, lakes, caves, tar pits, volcanic ash beds, amber and bogs, but the opportunities are much patchier. The best candidates for fossilisation are abundant organisms with durable hard parts, living in environments where burial is likely. The worst candidates are rare, delicate, entirely soft-bodied organisms living in environments where remains are exposed, destroyed or recycled before burial. The important implication of all this is that the fossil record is biased from the start. It overrepresents organisms with hard parts, organisms living in depositional environments, and organisms that were abundant enough to leave many chances for preservation. And it underrepresents soft-bodied animals, small delicate organisms, upland terrestrial communities, and behaviours or tissues that don't survive decay. If we focus just on marine organisms, fossilisation is still uneven. A burrowing bivalve, such as Pholus, has a relatively good chance of preservation because it's got a durable shell and it already lives buried within sediment. Epifaunal organisms, like the cementing bivalve Gryphea, also preserve well because they've got robust shells and they live in marine settings where sediments can accumulate. Pelagic organisms, those swimming around within the water column, have a different route into the fossil record. They live in the water column, not on or in the seafloor. But their remains can sink after death. Fish, ammonites, plankton, and other pelagic organisms can be preserved if they fall into settings where decay, scavenging and disturbance is limited. In very broad terms, there are 4 categories of body fossil preservation. And these are not mutually exclusive. The first is compression and impression fossils, where the organism is flattened into the sediment or leaves a surface imprint. These are especially important for plants and soft-bodied organisms. The second is permineralized fossils, where mineral-rich water enters pores or spaces in the original tissue and precipitate within them. This can preserve fine three-dimensional structure, especially in bone, wood, and some shells. The third class is casts and moulds. In these fossils, the original organism or hard part may dissolve away, but its shape is retained either as a cavity, which is the mould, or as a sediment infilling, which is the cast. The fourth class is unaltered remains. These are cases where original biological material survives with relatively little chemical alteration. Better known cases include material trapped in amber, frozen remains, mummified tissues, or some shells and bones retaining original mineral or organic components. And these are usually geologically young or preserved under extremely unusual conditions. So let's take each of those classes in turn. Compression fossils form when an organism or part of an organism is buried in sediment and then flattened as the sediment is compacted. Water is squeezed out, sediment grains are pressed closer together, and the original body is compressed into a thin layer. Now, these fossils are especially common in fine-grained sediments like mudstones and shales. Fine sediment is important because it can capture delicate outlines and surface details much better than coarse sand or gravel. And this is why compression fossils are so important for plants, algae, and soft-bodied animals and other delicate structures. In many cases, the original organic material is largely lost, but a thin carbon film can remain. This is sometimes called carbonization. The volatile components of the tissue are driven off during burial and heating, while a darker carbon-rich residue is left behind. In plant fossils, this can preserve leaf shape, venation, and fine surface detail. In animal fossils, it can preserve outlines of soft tissues that would otherwise decay completely. It's useful to distinguish compression fossils from simple impressions, although the two often occur together. A compression retains some flattened residue of the organism, like a carbon film. An impression is just the mark left in the sediment after the organism itself has gone. In practice, a fossil slab can preserve both, a film of organic material in some areas and only an imprint elsewhere. The second class of fossilization is permineralization. This is one of the important ways in which fossils preserve three-dimensional structure. It happens when mineral-rich water permeates the pores, spaces or cavities in the original tissue and precipitates out minerals. In wood, for example, it can move through the cells, vessels and spaces between cell walls. Minerals then build up in those spaces, strengthening the structure and helping it survive as a fossil. The minerals involved in permineralization include silica, calcite, iron minerals and phosphate, depending on the chemistry of the groundwater and the sediment. The process can preserve fine detail because the minerals can form before the original structure has completely collapsed or decayed, or even while the organism is still alive. Now, this is different from a simple cast. In a cast, the original object may dissolve away and leave a cavity that's later filled. In permineralization, at least initially, minerals form within the original tissue. A particularly important form of permineralization is silicification. A classic example is the Rhynie Chert from Aberdeenshire in Scotland. This is an early Devonian deposit, roughly 407 million years old, and it preserves one of the earliest known terrestrial ecosystems in fantastic detail. The Rhynie Chert formed in a setting influenced by hot springs and silica-rich waters. Parts of an early land ecosystem were rapidly entombed and mineralized by silica. That's why the preservation is so remarkable. In some specimens, plants are preserved three-dimensionally, and we can see conducting cells, sporangia, epidermal details and stomata. This is very important because early land plants were small and anatomically simple compared with later vascular plants, but they were central to the colonisation of land. Plants such as Rhynia, Aglaophyton and Asteroxylon show us early experiments in plant organisation. The Rhynie chert also preserves fungi, algae, cyanobacteria and arthropods, so it gives us a snapshot of an ecosystem rather than just isolated specimens. For example, the Trigonotarbids were extinct arachnids, spider-like in general appearance, but not true spiders. These animals are important because they show that terrestrial predatory arthropods were already part of these early land ecosystems. Rhynie chert fossils are often studied in thin sections, very thin slices of rock mounted on glass and examined under the microscope. That means we're often looking at a cross-section through a plant, an arthropod limb or part of the body, and a series of sections can be used to reconstruct the three-dimensional anatomy. Another important mode of mineralization is pyritization. In these fossils, tissues or cavities are replicated by pyrite, iron sulfide, sometimes called fool's gold. These fossils can be very striking because pyrite has a metallic, brassy appearance. Pyritization usually requires very particular chemical conditions: organic matter, iron sulfate, and low oxygen. As the organism decays in an oxygen-poor setting, sulfate-reducing bacteria can use sulfate in the surrounding water or sediment. One of the products of this process is sulfide. If dissolved iron is also present, iron and sulfide can combine to form iron sulfide minerals, including pyrite. The decaying organism is important here because it helps to create the local chemistry. It supplies organic matter for microbial activity, which creates a chemical microenvironment around the carcass. If pyrite forms early enough, before the tissues collapse, it can replicate fine details of the original anatomy. Carbonate mineralization is another important pathway. Here the minerals involved are usually calcium carbonate, such as calcite, or sometimes magnesium-rich carbonates, such as dolomite. Carbonate mineralization is common partly because carbonate chemistry is widespread in natural waters. Coal balls are a classic paleobotanical example. Despite the name, they're not balls of coal, they are carbonate-rich nodules found within coal seams, especially in Carboniferous coal deposits. They formed when peat, made up of plant material from ancient coal swamps, was permineralized before it was fully compacted into coal. A very different example of carbonate mineralization is the Herefordshire-Lagerstaette or Herefordshire fauna from the Silurian of the Welsh borderlands. These fossils are marine animals preserved in 3 dimensions inside nodules within a volcanic ash deposit. The preservation is unusual because many of the animals were soft-bodied or had delicate soft parts. They were buried rapidly, and as the soft tissues decayed, they left tiny voids. Calcite then precipitated in these spaces, producing 3-dimensional mineral infills of the animals. This means the fossils are not flattened impressions, they are preserved in the round. The Herefordshire fauna includes arthropods, worms, molluscs, echinoderms, sponges and other small marine invertebrates. Because the fossils are enclosed in hard nodules, they've often been reconstructed by serial grinding or sectioning. The specimen is ground away, layer by layer, each surface is photographed, and the images are combined digitally to produce a 3-dimensional reconstruction. Casts and moulds are another very common type of fossil preservation. They are especially useful because they can preserve shape even when the original material has completely disappeared. A mould is the hollow impression left behind when an organism or part of an organism is buried in sediment and then decays or dissolves away. The mould can preserve the outside of the organism, giving an external mould, or it may preserve an internal surface. For example, if sediment fills the inside of a shell and the shell later dissolves, the remaining stone records the internal shape of the shell. This is often called an internal mould or steinkern. A karst forms when that mould is later filled with sediment or mineral. In simple terms, the mould is the negative impression and the karst is the positive copy. The karst can look very much like the original organism, but it's not the original biological material. Calamites were tree-sized relatives of modern horsetails, common in Carboniferous coal swamp environments. Their stems were jointed and ribbed, rather like giant segmented reeds. Many fossils labelled Calamites are not preserved stems in the ordinary sense. They are karsts, often pith karsts, produced when sediment filled the hollow central cavity of the stem. The outer tissues of the plant then decayed or were lost, leaving the sediment infill as a fossil karst. The final category is largely unaltered remains. These remains have usually dried, frozen, tanned, oxidised, or lost some chemical components. However, compared with fossils that are compressed, replaced, permineralized, or preserved only as casts and moulds, they retain much more of the original biological material. The common factor is that decay has been slowed or inhibited. Ice slows microbial activity and chemical reactions. Amber seals small organisms away from oxygen and water. Bogs can be acidic, waterlogged, and oxygen poor. And caves can provide stable temperature and humidity and protect remains from weathering and scavenging. Amber is fossilised tree resin. It begins as a sticky secretion, especially as a response to injury, herbivores, or infection. Small organisms walking on the tree can become trapped in the resin. Insects are the classic example, but amber can also contain spiders, plant fragments, feathers, fungi, and even tiny invertebrates. The preservation can be astonishing because the resin surrounds the organism quickly and seals it from much of the outside environment. Small structures like wings, legs, antennae, and compound eyes can be preserved in fantastic detail. But amber has its own biases. It preferentially preserves small organisms associated with resin-producing forests, and it's not a general sample of an ecosystem. And although amber can make fossils look almost freshly dead, they are still fossils. The resin has hardened, polymerized, and changed over geological time, and the organism inside may be extremely detailed externally, but its internal tissues may be altered, collapsed, or degraded entirely. Bog bodies are usually better regarded as subfossils rather than fossils in the strict sense. However, they're still useful here because they illustrate a very different mode of preservation. Bogs are wet, acidic and oxygen-poor environments, often rich in sphagnum moss and humic acids, and these conditions inhibit many decomposers and can tan soft tissues, rather like leather. The result is not conventional fossilization by mineral replacement or impression, but the long-term survival of original organic material. And this is why skin, hair, nails, stomach contents, and other soft tissues can survive for long periods. Tolland Man from Denmark is a famous example. His face, skin, and other soft tissues are so extraordinarily well preserved that the body was initially treated almost as a recent forensic case rather than an archaeological find. However, this subfossil preservation is extremely selective. The same acidic conditions that preserve and tan soft tissues can dissolve bone and other mineralized tissue. So a bog body may therefore preserve skin and other soft parts while losing much of the skeleton. Tar pits give us another route to largely unaltered or unusually well-preserved remains. La Brea in Los Angeles is the classic example. Natural asphalt seeps to the surface and can trap animals that become stuck in it. The asphalt can protect bones from some destructive processes, but it also stains, impregnates and alters them. Brea is particularly valuable because it gives a remarkably detailed, though preservationally biased picture of late Pleistocene life in southern California. The asphalt seeps preserved huge numbers of bones, including mammoths, mastodons, saber-toothed cats, dire wolves, ground sloths, horses, camels, and many birds. However, this is not a perfectly representative sample of the whole ecosystem. Predators and scavengers are especially common, probably because they were attracted to animals already trapped in the asphalt. The La Brea fossils are not the result of one event. They accumulated over thousands of years, so that La Brea is a time-averaged record of many trapping events, changing environments, and repeated ecological interactions. It's worth treating microfossils separately. They are not really a fifth type of preservation because microfossil tells us about size, not process. Hence they cut across the preservational categories above. Coccoliths are tiny plates of calcium carbonate produced by coccolithophores, which are single-celled photosynthetic plankton living in the surface waters of the ocean. In life, these plates fit together around the cell to form a kind of spherical armour called a coccosphere. After death, the individual plates fall apart and can sink to the seafloor. Each coccolith is only a few microns across, but because coccolithophores occur in such huge numbers, their remains can accumulate on a vast scale. The White Cliffs of Dover are one of the classic examples. The chalk is made largely from the accumulated remains of coccolithophores and other microscopic plankton that lived in warm, late Cretaceous seas. So there's a striking contrast here between scale of cause and scale of effect. Minute calcite plates, far too small to see with the naked eye, built up into hundreds of metres of chalk across southern England and beyond. This makes coccoliths a particularly nice example because they show that microfossils are not just obscure microscopic details, they can form entire rock units and landscapes. Coccoliths are also important because they are useful stratigraphically. Different coccolith species evolved, appeared, and disappeared through geological time, and because they were widely distributed in the oceans, they can be used to date and correlate marine sediments. They're especially valuable in fine-grained rocks and deep-sea cores where larger fossils may be rare or absent. In addition, changes in coccolith assemblages can tell us something about ancient ocean conditions, including temperature, productivity, and broad climatic change. Foraminifera, or forams, are single-celled protists that build protective shells, more properly called tests. And these tests are often made of calcium carbonate, although some forams make agglutinated tests by cementing together sediment grains and other particles. Many forams add chambers as they grow, producing the beautiful spiral, coiled, globular or elongate forms shown here. Although the organisms themselves are microscopic, their tests are resistant enough to enter the fossil record in enormous numbers. They're important partly because they are so abundant. A small sample of marine sediment can contain thousands of foraminiferal tests. Some species live floating in the plankton, while others live on or within the seafloor sediment. This makes them very useful for reconstructing ancient marine environments. Different species are associated with different depths, temperatures, oxygen levels, nutrient regimes, and water masses. Their chemistry can also be analysed. Oxygen isotopes in foram calcite, for example, are used widely to infer ancient temperature and ice volume changes. Pollen is one of the most useful kinds of microfossil because, again, it's produced in huge numbers and has an unusually resistant outer wall. Strictly, palynology is the study of pollen, spores and other microscopic organic walled remains, while paleopalynology deals with these materials in the fossil record. Pollen itself is produced by seed plants, and spores are produced by groups including ferns, mosses and fungi. Both can preserve very well because their outer walls contain sporopollenin, a highly resistant organic polymer that's very difficult for most decomposers to break down. Pollen grains are not just tiny and durable, they are also morphologically highly distinctive. Different plant groups produce pollen with different shapes, sizes and surface ornaments. Some grains are smooth, some are spiny, some have pores and others have long furrows. This means that, under the microscope, fossil pollen can often be identified to a relatively narrow plant group. It gives us a way of reconstructing vegetation even when leaves, wood and seeds are absent. Pollen is especially valuable in sediments such as lake muds and peat bogs. By sampling successive levels, we can see how the pollen assemblage changes. For example, an increase in tree pollen might indicate woodland expansion, while more grass or herb pollen might suggest more open habitats. Changes in the relative abundance of different pollen types can therefore reveal shifts in vegetation, climate, and even human disturbance. So far, we've focused on body fossils, bones, shells, tissues, cast, moulds, and mineralized remains. But some of the most important and informative fossils are trace fossils, or ichnofossils. These preserve what organisms did, rather than the organisms themselves. Burrows are one of the most important kinds of trace fossil. This is a structure made when an animal moves through or lives within sediment. It can be a simple tube, a branching network, a feeding trail, or a more complex three-dimensional system. In some cases, the same animal can make different traces under different conditions, and different animals may make rather similar traces. So trace fossils are normally classified by their form and inferred behaviour, rather than by the exact biological species that made them. Burrows are particularly useful because they can tell us about soft-bodied animals that might never be preserved as body fossils. They can also tell us about the sediment itself. Horizontal burrows may suggest animals feeding or moving across or just below the sea floor. Vertical burrows can suggest dwelling tubes, suspension feeding, escape from rapid burial, or attempts to maintain a connection with the sediment-water interface. Dense networks of burrows indicate bioturbation, the churning and mixing of sediment by organisms. There's also an important evolutionary point here. The appearance and diversification of burrowing animals changed the seafloor profoundly. Once animals began to mix sediment more intensively, they altered oxygen penetration and nutrient cycling, and the preservation of sedimentary structures. So burrows are not just passive records of behaviour, they are evidence of animals engineering their environment. Footprints are another familiar and very useful kind of trace fossil. Like burrows, they don't usually preserve the body of the animal, they preserve behaviour. A single footprint can tell us about the shape of a foot, the number of digits, the presence of claws, for example, and sometimes even the way in which weight was distributed as the animal stepped. But a trackway is much more informative than a single footprint because it gives us a sequence of movements. From a trackway, we can estimate stride length, pace, the direction of travel, and sometimes relative speed. We may be able to infer whether the animal was walking, running, turning, or moving as part of a group. In dinosaurs, for example, trackways have been used to infer posture and herding behaviour, and the presence of animals in places where body fossils may be rare or absent. Coprolites are fossil feces. They sound unglamorous, but they can be extremely informative because they preserve evidence of food after it's passed through an animal's gut. They can contain fragments of bone or fish scales, shells or plant tissue, and these inclusions can give us direct evidence of what the animal was eating. They can also tell us about digestion. If bone fragments are etched, rounded or partly dissolved, That suggests strong stomach acids or prolonged digestion. There are, however, two major cautions. The first is identifying the producer. Unless the coprolite is found inside the body cavity of an animal, or very closely associated with a likely maker, it can be difficult to know which organism produced it. Shape, size, spiral structure, and the contents and context may all help, but they rarely give absolute certainty. The second caution is that coprolites are selective. They preserve what survived digestion, not everything that was eaten. The final category is chemical fossils, also called biomarkers. These are chemical traces of biological molecules that have survived, usually in altered form, within rocks or sediments. Living organisms contain biological lipids, such as pigments and membrane molecules. After death and burial, these molecules can be altered by heat, pressure, and chemical reactions. The original biological molecule may not survive intact, but a more stable derivative may remain. That derivative is the geolipid, or biomarker. Cholesterol is a biological sterol found in animal cell membranes and in related forms across eukaryotes. During burial and geological alteration, sterols can be transformed into more stable hydrocarbons called steranes. Cholestane is one such geologically stable derivative. Chlorophyll A is the familiar green pigment involved in photosynthesis. The original chlorophyll molecule is not usually stable over geological time, but parts of its molecular structure can be transformed into more stable compounds, including porphyrins. Porphyrins are useful because they retain a chemical relationship to biological pigments such as chlorophyll. Their presence in ancient sediments can therefore point towards biological sources of organic matter, including photosynthetic organisms. But biomarkers also need cautious interpretation. A molecule in a rock may have been moved, been contaminated, or indeed come from more than one source. So, as with trace fossils, the key is context. So, to form a fossil, especially a good fossil, the remains need to escape the normal processes that recycle organic matter. The first requirement is often an anoxic, or at least low oxygen environment. Oxygen allows rapid microbial decay and oxidation, so reducing oxygen slows some of the most destructive processes. The second requirement is protection from physical disturbance. If remains are exposed to water currents, waves, and scavengers, they're likely to be scattered, broken, or destroyed. The third requirement is rapid burial. Burial protects remains from scavengers and physical disturbance. It reduces exposure to oxygen and creates the chemical microenvironment in which mineralization can begin. Rapid burial doesn't guarantee fossilization, but without it, the odds are usually poor. The broader conclusion is that the fossil record is not a neutral archive of everything that once lived. It's a filtered record, shaped by anatomy, environment, decay, burial, chemistry, time, and later discovery. Understanding those filters is what allows us to use the fossil record properly, not just as objects, but as evidence for the history of life.